Good afternoon, everyone. The Biochemical Society and Portland Press are pleased to welcome you to this webinar, which is part of our Biochemistry focused webinar series. So topics in this series include different research areas in the molecular biosciences, as well as practical sessions to support career development. Each webinar will give you the opportunity to ask questions, and we welcome suggestions for future topics and speakers to feature in our webinar series. So please see the website for more details. I'm Dr. Vanja Pekovic-Vaughan. I'm a senior lecturer in circadian musculoskeletal physiology in the Department of Musculoskeletal and Aging Sciences at the University of Liverpool. Within the Biochemical Society, I'm a committee member of Research Area 6, which is Biological Systems. I'm also one of the network coordinators of the ECMAge network, one of the 11 UKAI-funded interdisciplinary aging networks, which bring together expertise in key aspects of aging, matrix biology, chronobiology, computational modelling, and tissue engineering across UK, and also offers free membership and pump priming and exchange funding to support a variety of projects. To learn more, please go to www.uknet.org.uk/ECMAge. Now, our speakers today are members of the ECMAge network, and I'm delighted to chair this webinar today entitled Extracellular Matrix and Redox Signaling Axis from Cancer to Tissue Repair and Smarter Regenerative Therapies. This is part of the Biochemistry Focus webinar series, a dedicated early career research program of webinars. We are therefore excited to provide you this opportunity today for several of our speakers, Mia, Tamara, Alexander, and Abhishek, to share their fascinating work within the molecular biosciences community. Before I hand over to our first speaker, I'd like to mention that questions will be asked at the end of the webinar, so please do send your questions during the talks as we go along. If you have a question, just type it in the question box as shown in the image on the screen, stating who your question is for, and we will try to answer as many questions as possible at the end. So our first speaker today is Mia Mohammed from University of Surrey. Mia is a final year PhD student at Surrey. She's undertaking musculoskeletal research in Dr. Giovanna Nalesso's lab. Mia has a background in biomedical sciences and a master's in stem cell and regenerative biology. After completing her PhD, Mia plans to continue her research on designing new technologies in tissue engineering. So today, Mia will present her work entitled Proteomic Insights of Sex and Age Differences in the Articular Cartilage. I can see your talk now, Mia.
Great.
Please go.
Thank you for that, Vanja, and thank you for this opportunity as well. So in today's talk, I'll be talking about our proteomic insights of sex and age differences in the articular cartilage. So the articular cartilage is a crucial tissue located at the end of our joints. This enables for efficient joint movement. It's made up of cells called the chondrocytes, and these cells constantly remodel the thick tissue matrix and gives it elasticity and resistance to pressure. The matrix is mostly composed of collagens, proteoglycans, and water, which all determine its mechanical properties. Factors like aging and sex can change the matrix by affecting its structure and function of the tissue, and these changes may lead to osteoarthritis, a chronic joint disease that affects 73% of people over the age of 55 worldwide and is more common in females. However, despite its high occurrence in women, only 42% of the studies consider gender differences, which highlights the need for more research in this area. So in order to fully understand this association, our project aims to understand young and old sex differences of the articular cartilage extracellular matrix for a better translational understanding of aging and predisposition of OA. First, we performed a proteome analysis of the extracellular matrix isolated from old and young mice. This involved 12 murine articular cartilage samples consisting of 3 old females, 3 young females, 3 old males, and 3 young males. These samples then underwent tandem mass tag mass spectroscopy, which is a labeled quantitative proteomic approach that allows the comparison of protein levels in multiple samples using specific mass tagging, and then the label samples are combined prior to the subsequent processing steps, which eliminates run-to-run variability, which can be an issue when samples are analyzed consecutively. And then the data was followed by bioinformatic analysis. The initial set of analysis then was carried out, and this involved a principal component analysis. So this plot accounts for the variance between samples and triplicates of the samples. So from this plot, we can see 3 out of the 4 sample groups are shown to be consistent across the triplicates, as their plots are close in proximity. However, as you can see as a whole, you can still observe 4 whole protein groups being old males, young males, old females, and young females. In panel B, we found 2,484 proteins which had a high FDR, false discovery rate, of 5%, were identified across the 4 groups, and we observed 12% of those proteins were extracellular matrix proteins, which were manually identified using UniProt database. This was then confirmed in a gene set enrichment analysis, as extracellular matrix related pathways were identified to be downregulated in the ages in I and II, whereas in III and IV, we can see that the aging sexes showed opposite trends, which displayed the same pathways, which were upregulated in the aging males and downregulated in the aging females. So overall, these analyses determined that there are differences between the aging sexes in relation to cartilage ECM remodeling. Next, we showed differential modulation of these identified extracellular matrix related proteins using volcano plots. So these plots were able to show the specific proteins that were significantly modulated in each comparison group. And as you can see, there's more significant proteins identified when comparing the different sexes at the same ages, so the last 2 volcano plots. And this supports our findings from the last slide. We also conducted a string protein-to-protein interaction analysis that shows how these modulated proteins interact with each other. So this mostly displayed down or upregulated protein trends were interacting with each other with similar modulated proteins. And then this can further help identify the ECM related pathways that they are affected in. Additionally, thanks to the Pump Prime ECMAge funding, we were able to go deeper into the proteomic analysis as we identified structure associated differences of those ECM related proteins using peptide location fingerprinting. So in the collaboration with the University of Manchester, we adapted their tool for evaluating age and sex-related structural modification in murine articular cartilage proteome using tryptic peptides acquired from the tandem mass spec analysis. So this tool works by dividing the protein's primary sequence into 50 amino acid segments, which are then quantified using their abundances and then mapped to the respective segments. So here in 2 is a line graph showing the normalized peptide abundances comparing the young and old cartilages of both sexes. And beneath you can see the amino acid segments aligned with the domains and regions that they are part of. So in our analysis, we compared young versus old males and females, as well as young males and females versus old males and females. So as you can see, this PLF analysis established more than double the number of significant proteins, showing structure-associated changes than we had previously measured using the abundances values alone. So as an example of one part of the analysis, we categorized the proteins using Panther classification system and found 27 extracellular matrix proteins, which were further separated into structural proteins associated with matrix proteins and proteoglycans. So the PLF analysis was able to identify the structural associated differences along the proteins. So as an example, in Col15 alpha 1, a protein which stabilizes the extracellular matrix, you can see a conserved modulation of the structure associated differences between the male and females. And here in laminin G-like domain, there's a higher peptide yield in both of the sexes. This is expected as this domain influences cell adhesion, which is an important characteristic of the tissue. The further down you go, you can see higher peptide yield in the triple helical region, of the old, which implies the alteration of a higher order structure of the protein. A sex-divergent structure-associated difference was shown in a pericellular matrix-located protein, collagen 11 alpha 2. The fibrillar collagen NC1 domain shows a higher peptide yield in the young females and old in the males. This domain is susceptible to proteolytic cleavage, which could change the function of protein as a whole. And as a whole of this analysis, it helps identify sex differences, which would be great for potentially identifying sex-specific targets for development of targeted therapeutic protein interventions. So for our first experimental application, which was also funded by the ECMAge pump priming in collaboration with the universities of Oxford and Southampton, these experiments involved identifying a protein that was differentially upregulated in aging, but was conserved between the genders using the mass spec data we had gathered. We established collagen 5 alpha 2 to be further assessed for its impact in aging of the extracellular matrix. This protein has been previously associated with genetic mutation in COL5 in Ehlers-Danlos syndrome, which among its symptoms include hypermobility and skin that appears prematurely aged. But also in this publication, this gene has been proposed as a biomarker of the maturity of aging of the human articular cartilage. So with collaboration with our universities, we decided to generate a young in vitro cartilage model using gelatin methacryl to assess the phenotypic effect on human articular chondrocytes. And this model is currently undergoing characterization, but however, we have seen promising initial results as this Fickrix-Cyrus red staining shows an increase of stain intensity from day 4 to week 3 of cultivation. So this shows that the gel can accommodate the cells as there's an increase of collagen production. Overall, we believe this data provides a significant insight to the sex-related differences of the articular cartilage, and using the same tool, similar data can be acquired for other tissues and disorders. Also, we have a better understanding of sex differences in the cartilage biology, which can help contribute to smarter personalized treatments for cartilage-related diseases. And finally, finally, our proteomic analysis has acquired a huge amount of normal data, which could pave a basis of many future research. So I would like to take this opportunity to thank my funders, Dunhill Medical Trust and ECMAge, our collaborators with the University of Manchester, Oxford and Southampton for helping us to excel in our research and determine novel findings. And finally, I would like to thank my supervisor, Giovanna, and our research group for all the support, motivation, and inspiration you have provided me in my journey. Thank you all for listening.
Thank you so much, Mia. That was a great talk. Please put in some questions for Mia's talk, and we will discuss this at the end of our webinar. Now for the next talk, our invited speaker is Tamara Zakic from University of Belgrade. Tamara has a master's degree in molecular biology and physiology and is currently a PhD student at the Institute for Biological Research, University of Belgrade, in Professor Bata Korac's lab. Tamara's PhD thesis focuses on redox and metabolic reprogramming in breast cancer, and today she will present her work entitled Redox Profiles as the Pivotal Features of Tumor Adipose Tissue Cooperation in Breast Cancer Patients. Over to you, Tamara.
Thank you, Vanja, and thank you for this opportunity. As Vanja said, the topic of my talk today will be redox profiles as the pivotal features of tumor adipose tissue cooperation in breast cancer patients. Breast cancer is the most common malignancy among women worldwide, where 1 in 8 women will be diagnosed with breast cancer during their lifetime, and except gender, there are other risk factors that affect breast cancer, such as age, personal history, family history, reproductive history, or some lifestyle-related factors such as obesity. The relationship between obesity and breast cancer in postmenopausal women is pretty much well known, and around 30% the risk factor for breast cancer is increased in obese women, postmenopausal women. However, this relationship is not very well known in premenopausal young women, since there were many conflicting results, some of them showing positive, some of them negative effects of obesity and breast cancer progression. This question is really important, since women diagnosed at young age have lower survival rates and they experience poor quality of life, since they're often diagnosed at a very later stage of disease. So understanding the mechanisms that underlie this relationship between neoplastic transformation and obesity in premenopausal women is of great importance. We could consider breast cancer as a complex pseudo-organ due to many reasons. Behavior of breast cancer cells alone depend on their interaction with the tumor microenvironment, its stromal components and extracellular matrix. Specifically, in breast cancer, the main component of the tumor microenvironment are cancer-associated adipocytes, and they contribute to cancer progression, metastasis, and therapeutic resistance by supplying breast cancer cells with growth factors, adipokines, cytokines, metabolic substrates, and this communication is a two-way communication, since they both affect on each other, which has been shown in many studies. However, the precise role of cancer-associated adipocytes in establishing breast cancer phenotype, and specifically the redox profiles of both breast cancer and cancer-associated adipocytes, are still under question. So we wanted to examine the hallmarks of neoplastic transformation reflected in redox reprogramming in both breast cancer and cancer-associated adipose tissue in premenopausal breast cancer patients. We had both paired biopsies of tumor and adipose tissue, which were divided by pathohistological analysis. Tumor tissue was divided into benign fibroadenoma, as well as into malignant type A invasive ductal carcinoma, while adipose tissue was divided into breast adipose tissue or cancer-associated adipose tissue. Women were divided by body mass index into normal weight and overweight or obese women, making 4 groups total: normal weight women with benign tumors, overweight obese women with benign tumors, normal weight with malignant, and overweight women with malignant tumors. Firstly, we examined the redox profile of breast cancer tissue. We observed increased protein expressions of the main antioxidant defense enzymes in malignant tumor tissue compared to benign tumor tissue, irrespective of obesity. Next, we wanted to see to indirectly assess ROS levels by measuring 4-hydroxynonenal protein levels as well as immunofluorescence, which showed no significant differences between the groups. These results point to the high redox threshold that is present in breast cancer tissue, which was already reported, and it is in accordance with previous results. And it actually does not point to increased oxidative stress, just to the increased redox threshold. Additionally, we examined the protein expression of NRF2 as the main transcriptional regulator, and we see its protein level increase only in obese women with malignant patients. This increase could be explained since the new role of NRF2 in metabolic reprogramming is recognized, and it is well known that obese patients have altered metabolism. So we can see that the redox response of breast cancer tissue is actually here not associated with obesity. Unlike tumor tissue, we examined cancer-associated adipose tissue that displayed increased protein expression of main antioxidant defense enzymes only in normal weight women, and this was in accordance with the protein expression of NRF2, pointing to its role in the regulation of redox reprogramming of cancer-associated adipose tissue. Additionally, we measured the 4 hydroxynonenal modified proteins, their protein level and immunofluorescence, where we observed increased levels only in obese women with malignant tumors. This was also expected since obese women are known to have increased pro-oxidant state along with the reduced antioxidant capacity, which was shown with expression of antioxidant defense enzymes. Since the tumor microenvironment of breast cancer is heterogeneous, we wanted to examine cell-specific localization of antioxidant defense enzymes in both cancer tissue and adipose tissue. We observed increased expression of antioxidant defense enzymes in malignant tumor tissue compared to benign tumor tissue, just like our Western blot results, and their specific localizations in either mitochondria or cytoplasm. We also examined regional differences in the expression of these enzymes, where we observed that clusters of cancer cells at the invasive front were much more immunopositive for antioxidant defense enzymes than in cancer cells in tumor core. We did the same analysis on the cancer-associated adipose tissue, where we also observed their increased expression in women with malignant tumors. They specifically localized, just like in cancer cells, and the expression of these enzymes increases with proximity to cancer cells. We here confirmed that there is a high redox homeostasis threshold in both cancer tissue and cancer-associated adipose tissue, and their responses, redox responses, are synchronated. And finally, we examined serial tissue sections on light microscope, where we observed only in malignant tissue, and especially in obese patients, that there is a physical association between these two tissues, with extracellular matrix serving as a connecting tissue between the two. We can draw a few conclusions from these results: that breast cancer and its associated adipose tissue exhibit synchronous tissue responses when it comes to redox reprogramming, that their mirroring phenotypes reveal tissue-specific high redox homeostasis thresholds. Their complementary differences points to their dependent cooperation, and that obesity might change the redox interface of cancer adipose tissue crosstalk. Finally, I would like to thank my team from the Institute for Biological Research Siniša Stanković, led by Professor Vato Korać, and our colleagues from Faculty of Biology, Faculty of Medicine, and Institute of Life Course in Medical Sciences, and thank you all for your attention.
Tamara, thank you for a lovely talk. We will now go to our next speaker. Our third speaker is Alex Eckersley from University of Manchester. Alex is a new lecturer in skin health at Manchester, funded by Walgreens Boots Alliance. Alex is developing novel mass spectrometry tools for identification of extracellular matrix targets in aging and connective tissue diseases. He has contributed to the design of new matrikine peptides used in the skin cosmetic Future Renew product line launched by Boots in 2023, and has also developed in vitro human skin cell culture assays for multi-omics characterization. This has all led to 4 patents where he's a named co-inventor. Today, Alex is presenting his work entitled Matrix-Derived Peptides for the Regeneration of Photo-Aged Skin. Over to you, Alex.
Apologies, is that better? Can you hear me okay now?
Yes.
Perfect. And my screen's showing?
It's
Great.
showing, thank you.
Excellent. So thank you very much to you, Vanja, and to the rest of the organizers for giving me the opportunity to present today. I'll be talking a little bit about a large collaborative project we've been running over the last 6-7 years together with our industrial partners at Walgreens Boots Alliance. So the first important thing to note is that ECM components are markedly long-lived. For example, only 80% of collagen II turns over in cartilage in an average 80-year-old. And as far as we can tell, elastic fibers do not turn over. So the ones that are there from birth remain present throughout your lifetime. This means that ECM components like elastic fibers can accumulate damage over time through repeated exposure to aging mechanisms like reactive oxygen species, ROS, proteases, and UV in photoexposed skin, which can lead to fragmentation and degeneration of their architectures. So the first question becomes, are we able to predict which skin ECM components are susceptible to these damage mechanisms? When it comes to UV and photodynamically induced ROS, our labs have shown that susceptibility to these mechanisms scales with the content of certain UV ROS absorbing amino acids, namely cysteine, methionine, tryptophan, and tyrosine, meaning we can predict UV ROS susceptible ECM proteins based on the presence of these residues. Protease susceptibility, however, is harder to predict as it relies on higher order structure as well as amino acid sequence. There are machine learning algorithms capable of predicting this, like Prosper, for example. However, we found that this didn't work very well with ECM, and so together with our colleague Matthijs Ozels, we developed our own called the Manchester Proteome Susceptibility Calculator, which worked much better when it came to when we validated it experimentally. This means that we can predict protease susceptibility on proteins based on their cleavage site load, and even the fragments that are yielded as a result of this cleavage. So why is it important to predict ECM protein damage and fragmentation? Well, that's because some ECM fragments called matrikines are able to modulate cell behaviors like migration, apoptosis, proliferation, meaning that some matrikines have the capacity to drive tissue repair and maybe even ECM deposition. And indeed, matrix-derived peptides are currently used within the anti-aging cosmeceutical industry with some proven efficacy. However, there's no clear hypothesis that drives new discovery of bioactive peptides. So we hypothesized that small bioactive peptides, also known as matrikines, that are capable of skin regeneration, can be predicted via in silico digestion of dermal ECM proteins through the action of proteases. So we selected 27 target proteins based on their predicted UV ROS and protease susceptibilities, and they're being previously reported as affected in aging skin. We use our machine learning algorithm to predict the cleavage products of these proteins by relevant ECM cleaving proteases. And this process alone led to around thousands of possible peptides, which we filtered down to 453 based on the length. We only wanted 4 to 5 amino acid long peptides, which are capable of penetrating skin more readily. Our partners at Sederma then filtered those based on their manufacturability and stability. So this resulted in a final list of 8 ECM-typic tetrapeptides, which could be synthesized by our industrial partners, all of which came from a diverse source of ECM proteins, including collagens, ECM glycoproteins, etc. So now that we had our 8 peptide matrikine candidates, we wanted to know if they could enhance deposition of key ECM markers in primary hemodermal fibroblasts, and importantly, if they exhibit differential effects on cell activity. And this work was carried out by our PhD student, Nathan, who has since passed his viva, I'm happy to say. So the first thing we found through immunofluorescence analyses was that all 8 peptides do enhance the deposition of key ECM markers when exposed to skin fibroblasts in vitro, but interestingly, they had differential effects, with some, such as P1 and P7, significantly increasing fibrillin 1, and others, like P4 and P5, increasing procollagen 1 and fibronectin, for example. So this shows that depending on the target sequence, these matrikine peptides seem to target unique processes for ECM deposition. We also extracted the matrix and secretomes from peptide-treated dermal fibroblasts and analyzed them by proteomic mass spec, and again found that they each had differential effects, with some, like P1, enhancing multiple ECM deposition and tissue generation protein classes, red being enhanced in the bubble plot, whereas others like P7 suppressing many of these classes. So it's clear that all 8 peptides are capable of modulating ECM deposition by skin fibroblasts in different ways, but we wanted a deeper understanding of their potential for regeneration. So we selected 2 out of these 8 peptides for further study. Peptide 1, which is GPKG amino acid sequence, and P7, which was the LSVD amino acid sequence, which were chosen because their sequences were found in several ECM components, and because they're both capable of enhancing ECM deposition, both on the marker and proteomic level. So to help define the effect of these 2 peptides, we incubated primary fibroblasts with 3 different three different skin sites for 12 hours with our peptides, extracted their total RNA and analyzed them by RNA-seq. And the first thing we found by principal component analysis, which was no surprise, was that the transcriptome clustered by site, as in where the skin came from. But if we do look at the individual PCAs, we can see that these peptides are leading to large changes in transcriptional activity. And indeed, we found these peptides enhance gene expression of pathways pertinent to ECM remodeling, as well as cell proliferation and even ROS response. We ran a parallel proteomic analysis of the secreted matrix, and this also showed shifts in matrazome profile following peptide treatments. We next wanted to know whether these peptides could act synergistically on dermal fibroblasts, and so we treated cells this time from the same site, buttock, with a combination of the two peptides, and found that they had a strong effect on the transcriptional activity in the secreted matrazome of dermal fibroblasts. As strong, in fact, as our positive control, all-trans-retinoic acid, which is a vitamin A derivative and one of the most researched bioactives used to rejuvenate photoaging skin. You can see that the combination treated group clustered nicely compared to the control, and even against the retinoic acid, showing that peptides have a strong but differential effect, even to the positive control. And like the individual treatments, we found that peptide combination enhanced transcriptional pathways pertinent to ECM remodeling as well. Perhaps most importantly, we found that the combination of peptides 1 and 7 led to a higher number of upregulated ECM-associated proteins compared to individual peptide treatments, highlighting the fact that these peptides really do act synergistically to enhance ECM production, at least in vitro. So we know that these peptide matrikines show promising activities in vitro, especially in combination. And of course, the next step is to test this in vivo. So our industrial partners at Boots created a topical formulation containing our combination blend, which we assessed in skin patch tests of human volunteers, which was carried out in collaboration with Professor Rachel Watson and Doctors Abby Langton and Bez Mamboy here at the University of Manchester. We applied it to photoaged forearms of 8 healthy individuals for 12 days. The treatments were compared to an included control, a vehicle control, which is made up of the cream, excipient but without the peptides, and also against an all-trans-retinoic acid positive control. Punch biopsies were then taken and analyzed by immunohistochemistry for fibrillin microfibrils and for transcriptomics as well. So if you think back to the first slide I showed, the elastic fibers, which are made up of fibrillin microfibrils and elastin, are the first components to degenerate in photoaging skin. And you can see in our control photoaged forearms a lack of fibrillin fluorescence in the papillary dermis. Encouragingly, we found that the peptide combination treatment led to the new deposition of fibrillin microfibrils in this region, and that this was significantly different, as effective as our positive control, all-trans-retinoic acid. Our transcriptomic analysis showed that exposure to the peptide formulations not only affected bulk changes in gene expression in skin, but also the enhancement of biological processes crucial to skin regeneration, including the epidermal maturation of keratinocytes, development of skin barrier, expression of ECM components, elastic fiber deposition, as well as modulation of biomechanical pathways pertinent to biomechanical properties. So finally, I just want to say that these peptides, peptide 1 and 7, are now, they've been incorporated in Boots' new number 7 product line launched last year, Future Renew. So literally, this collaboration project going from in silico to in vivo are now launched in an anti-aging cream, which I think is pretty amazing. This is, of course, made possible by Professor Mike Sherratt, who led the collaboration, and our partners at Boots, Dr. Mike Bell and Dr. Ellie Bradley. And this is just to say thank you to the whole team, both at University of Manchester, WBA, so that's Boots, and Sederma for making this work possible. Thank you very much.
Thank you, Alex, for that fascinating talk, and I'm sure our attendees will be keen to put some questions in question box about your work and the future of this work. And now for the next speaker, which is our 4th and final speaker today, is Abhishek Mahajan from Clatterbridge Cancer Centre, Liverpool. Dr. Mahajan is a consultant radiologist at the Clatterbridge Centre, Liverpool, with special interest in head and neck, thoracic and neurological cancers. He's been an active contributor to the field of AI, head and neck cancers, neuro-oncology and oncology literature by authoring hundreds of publications, several book chapters, and also has written and edited a textbook on oncological imaging. Today, he will present his work entitled Imaging-Based Assessment, Sarcopenia and its Implications on Outcomes in Oncology. Over to you, Abhishek. Thank you. I can see your slide now.
Oh, thank you. Thank you for the opportunity to speak at this webinar. I think I'm just the odd man out here, which is probably speaking on something which we do in the clinics and how we assess sarcopenia and its implication in oncology. So to give a background, sarcopenia is an important subject in the current field of oncology because it not only predicts outcomes, but also assesses whether the patient can handle the chemotherapy and radiotherapy. Patients who are actually sarcopenic tend to do worse in terms of progression-free and overall survival, as well as response to therapy compared to the patients who are normal weight with non-sarcopenic. And something which we now commonly come across is sarcopenic obesity. That means the BMI is normal or maybe obese, but their muscle quality is very poor. So what we understand from sarcopenia is it's a syndrome characterized by generalized loss of skeletal mass and also the strength of the muscle, which actually denotes the body composition and also reflects on the nutritional status of the patient. So as per the European consensus guidelines, the pre-sarcopenic patients are the one who have loss of muscle strength. Once the muscle quantity and quality starts to deteriorate, that kinds of leads to sarcopenia. And once there is loss of performance, that means the patient is unable to do day-to-day activities. That is severe sarcopenia. So this is what we try to analyze in our clinics. We assess the BMI, we look at the nutritional status, we try to upregulate the nutrition to see whether the patients can do better during the therapy and in terms of overall survival. However, primary sarcopenia, which is related to aging, can add on to the cancer-related secondary sarcopenia and make the patient very cachexic. So what happens in the oncology is we see that there are certain factors which you guys would be much more aware that tumor necrosis alpha factor interleukins, which acts on the muscle, leads to lipolysis, thermogenesis, and browning, which actually is the causative factor behind loss of muscle mass, increased visceral obesity, and increased visceral fat. That in turn leads to cancer cachexia. And once these cancer cachexic patients receive chemotherapy, they have higher incidence of GI toxicities, they have higher myolysis, their response to chemotherapy is less, and they have higher incidence of chemo-related overall toxicities. That reduces the amount of chemo cycles given to a patient. That means a patient who's sarcopenic may not receive adequate number of chemotherapy cycles. And if the sarcopenia is not corrected, a full treatment may not be provided to the patient, and that adds on to the overall survival of the patient. Keeping in this mind, we explored way back in 2015 that how could we evaluate the sarcopenia on imaging, which could play as a surrogate marker at the time of staging of the patient to understand whether these patients can be subjected to additional support with nutrition benefits supplements. And we found that the patient BMI wasn't a true representative of the overall nutrition status. As seen in these two patients, you can see that the patient's BMI in these two cases was near comparable. However, the muscle quality in the first patient is very poor. You can see a lot of green areas, which represents the fat within the intramuscular septum. So this intramuscular fat increase shows that the muscle quantity is less. And also within the muscles, if you see the density of the muscle is low, that is a skeletal muscle density, which indirectly represents the quality of the muscle. So the quantity is less, the quality is less, and then hence the overall performance of the muscle is less. Compared to the other patient who had the same BMI of 26.8, however, there is hardly any intramuscular septal fat. The muscle quality is very good, the skeletal muscle index is very good, and this patient is a non-sarcopenic patient. Comparing the two, this patient would have done better in terms of response to chemotherapy, toxicities related to chemotherapy, and overall survival. With this hypothesis in mind, we looked at the effect of body composition on CT scan and comparing the outcomes in patients with esophageal cancers. And what we looked at was the skeletal muscle density and overall skeletal muscle index. What we found, apart from the most important factor, which predicts the outcomes in patients, that is lymphovascular invasion, the skeletal muscle density was an independent factor for predicting the response. So patients who had better skeletal muscle density, that means the muscle were more denser, did better than the patients who had poor skeletal muscle density. We further went on to exploring the inflammation related to cancer, and this is commonly known as cancer inflammation index, which is represented by the neutrophil to lymphocyte ratio and the serum albumin levels. So normally when you calculate a inflammatory index, you use PMI into serum albumin divided by neutrophil to lymphocyte ratio. What we did, we substituted the BMI with the skeletal muscle index, as we found that was a better predictor of the outcomes, and compared the overall the progression-free survival and the number of toxicities. And we found that the sarcopenic patients did worse than the non-sarcopenic patients in this cohort, and also the patients who had the progression-free survival also in these patients were better than in terms as compared to the non-sarcopenic patients. Apart from the sarcopenia, the skeletal muscle density was also an independent predictor of progression and free and overall survival, and hence was the modified inflammation index. Similar results have been published in the literature where the researchers have found that the sarcopenic patients in hepatocellular cancer tend to do the pink denotes the sarcopenic patients and the blue denotes the non-sarcopenic patients. The non-sarcopenic tends to do better than the sarcopenic patients. And in terms of toxicity, again, it has been shown that in colorectal cancer, the non-sarcopenic patients had lesser amount of toxicities, that you can see 2 out of 15 as compared to the sarcopenic patients, and the p-value was significant. With this, there is a recent meta-analysis and systematic review which was published, which showed that the sarcopenic patients who have lesser muscle strength and quality tend to do worse in terms of overall survival, progression-free survival, and quality of life index. So with this hypothesis, what we started to do at Clatterbridge is, this is the normal pathway. We do the initial workup for the patient. We look whether the patient is of curative intent or palliative intent, and hence the patient is offered surgery or preoperative neoadjuvant chemotherapy or postoperative neoadjuvant chemotherapy. However, now what we do is we look at the baseline imaging of the patient, evaluate the sarcopenia, and if the patient is sarcopenic, we give nutritional supplement, correct the sarcopenia, and also support the nutrition supplementation during the course of the treatment and after the course of the treatment with high protein diet. This has led to better outcomes in our clinical cohort. Now, the way forward to look at this is to develop automated tools using machine learning to assess sarcopenia, because calculating imaging-based sarcopenia is still a time-consuming process, and not all radiologists are trained to assess this. And second is to delve into the molecular mechanisms of multiomics in cancer-related sarcopenia. There is something which is recently published, which is detection of sarcopenia using deep learning-based body part measure system, that is the AMBI, which automatically calculates these SMI, that is skeletal muscle index, and calculates the sarcopenic index as well. And recently I've come across this paper which talks about prospective role of liquid biopsy in sarcopenia to assess and compared with the imaging based sarcopenia, and has shown to play quite an important role in the clinics. And liquid biopsies are commonly available for these patients because they are part of the biobanks. So various pathways which are beyond the confines of my knowledge have been tested in these, and they have been found to be related to sarcopenia in oncology patients. The idea for this topic was to build up collaborations with the people who are working in the lab, and if there is any future prospects to do a joint work with somebody who's interested in this from Liverpool and across. With that, I thank you for your patient hearing. Any questions, I'll be happy to take.
Abhishek, thank you very much for that great talk. Now we welcome questions for any of the speakers. So please, if you have a question, type it in a question box as shown on the image on the screen, and then we can have the next 10 to 15 minutes having some questions for the speakers. Okay, so I have a question here from the attendees for the first speaker, Mia. So for the recombinant COL5A2, is this a single long polypeptide, or is it expected to trimerize? And also, would it stay intact, or would it be converted to active fragments? Over to you, Mia.
Thank you for the great question. So it's not as long as the other collagen strands, such as COL2 and COL1, but it is prone to fragmentation because it also has the NC1 domain, so it can fragmentize. And I think also there's a different chimeras of it as well. I don't know too much into it because it has looked into the Ehlers-Danlos syndrome more in depth. Whereas in relation to aging of cartilage, it's only like 1 or 2 simple papers into it, and usually it's in relation with interactions with other collagen molecules or other extracellular matrix related molecules. So it would be something that we can specifically look further into, especially with peptide location fingerprinting and other analysis in vitro as well.
Thank you, Mia. I also have a question for you. So would you be able to tell us a little bit about, so what are some of the challenges you experienced looking at the proteome and extracellular matrix from these young and old tissues, and some of the challenges you might have experienced with mass spec analysis?
So the main challenges was actually at the beginning, like just getting the tissue. So the big thing I noticed a difference with is getting the articular cartilage off young tissue was a lot easier. It slid off the femoral head very easy. Whereas when it came to the old tissue, it was quite anchored solid onto the femoral head. So it meant that we had to break them in pieces, or alternatively, like use a decalcification solution to get rid of the bone and then take the cartilage aside for the proteomic analysis. And as I've never done any proteomic analysis before this, there was a lot to learn and understand. So yeah, I had the help of Alex. So he was one of my collaborators with the PLF. And also other help from the university of understanding what approaches, where to start off with and how to further go down in the line of what is the importance, because there is so much data you can get out of it. And yeah, just finding the key one that I want to stick to, so the extracellular matrix, and then finding what pathways I want to go down. Eventually, with all the learning and then all the training, I was able to overcome these challenges.
Thank you, Mia. That sounds so. I really like a very determination pathway there for you to get to those samples. Could you also just tell us? You mentioned that there was differences between obviously sex and age, and there were some, for example, targets that were upregulated in males and downregulated in females. So could you tell us some whether there were any specific sub pathways within extracellular matrix proteome that were of interest there, or is that something you're still sort of working out?
So in the gene set enrichment analysis, it was mainly just like a whole topic of ECM remodeling, so showing that there's collagen being synthesized, collagen being broken down. So this whole ECM remodeling pathways that were related and found in the aging. But one big thing we found when we were seeing the age, like young versus old in the specific sexes, we noticed that there were opposite trends in the female and males, which was an interesting thing to see because I think from what I remember in males that they were actually upregulated. So that means there was more remodeling going on, whereas this process was downregulated in females. And this might indicate why there is sex differences and what we should look at differently, and how we can kind of find specific targets in those pathways using the volcano plots that I showed and the protein to protein interactions to find exactly what pathway you would want. So I was just doing the whole overall proteome analysis, and then if we do want to look specifically into one, we will just go into depth and find one.
Well, thank you, Mia. That's great. So whilst I'm waiting maybe for some more questions from our attendees, I'll go to our next speaker and ask her a couple of questions. So this was Tamara. So Tamara, you showed us an interesting work about redox reprogramming between breast cancer tissue and cancer-associated adipose tissue. So tell us a bit more about how do you explain these really interesting differences in NRF2 expression level, one of the key antioxidant markers, between, for example, breast tissue and adipose tissue, and do you think that maybe NRF2 plays a different role in these tissues? And is there much work out there linking the NRF2 levels to different levels of weight and obesity?
Okay.
Thank you.
Thank you for the question. Here we just measured the protein expression of NRF2, and that could be one of the explanations for the differences in breast tissue and adipose tissue, since we didn't measure the actual activity or the nuclear expression of NRF2. So that might be one of the reasons why they differ in adipose and breast tissue. And there are many papers on the role of NRF2 in cancer, some of them pointing its positive or negative effect or helping in breast cancer progression. Some of them confirming the other. Regarding the role of NRF2 in adipose tissue, there are not so many results. So this protein expression of NRF2 is just the beginning of seeing its role in adipose tissue. And as far as cancer tissue is concerned, it's still under debate, and it's different in different tumors and in different types of breast tumor. Its expression and its activity. So I think definitely the further studies are needed to determine its exact role, specifically in breast cancer tissue.
Thank you, Tamara. That sounds great. So I have here a question from the attendees for Abhishek. So the attendee would like to know whether and what nutritional interventions composed of different aspects and for how long might be needed to correct sarcopenia in cancer patients prior to treatment.
Yeah, so prior to, I would say 2018, we didn't have a nutritional supplementation or available for patients, and patients were not referred to the nutrition department. But now, based on the imaging, each patient at Clatterbridge is defined as sarcopenic or non-sarcopenic. Higher priority is given to, because there is limited number of slots available in NHS, high priority is given to patients who are severely sarcopenic. The nutrition supplement can be through per oral, which is directly if the patient is able to swallow. We give protein based and vitamin D based high protein diets. If the patient cannot swallow, we put nasogastric tubes for these patients, and also for cancers such as head and neck cancers where patient is unable to swallow, we preamp the situation and put nasogastric tubes and improve this give supplementation, improve the nutrition, then start the therapy rather than directly jumping onto the therapy. Some patients we find even difficult to put a nasogastric tube, such as esophageal cancers, where the tube may not pass. So what we do is we puncture a hole into the anterior abdomen, put a peg which is direct communication with the stomach, and start giving nutrition supplementation through the stomach to the patient. And what we've seen is the failure rate and has reduced, and patients tend to do more cycles as compared to the patients who have not received nutrition supplement. Following our research, what we've tried to do is we are now focusing more on high priority patients than the less priority, because we do not want to lose the patients who are severely sarcopenic.
Thank you, Abhishek. So I have just a simple question for you. So obviously with the loss of muscle mass, you mentioned some of the things that you also find associated with it, like browning of white adipose tissue, some inflammatory markers and myosteatosis. So do we know what happens to the connective tissue, like the extracellular matrix, the collagens, and so on?
I honestly have no clue about it. I haven't come across any literature on that. What about the extracellular matrix? I'm sure there would be some changes into the vimentin pathway, which have been related to metastasis, would be one of the causative factors for poor prognosis. I just had a comment about Tamara's. I've done some research on breast adiposity and related to outcomes independently through mammograms. And what we found was, if you go back and look what kind of tumors you were dealing with, whether they were estrogen and androgen progesterone receptor positive or negative, that will give you an independent outcome, because the receptor positivity is directly linked to the hormone production and the tumorigenesis. And the obesity is indirectly related to outcomes because of sarcopenia, because most of these patients are sarcopenic obese rather than healthy obese. If there are some groups who are looking at actual patient cohort, I'm happy to help them with calculating sarcopenia on the images.
Great, thank you, Abhishek. And so please, attendees, if you have any questions for any of the speakers, do put it in a question box, but I'll also ask a couple of questions to Alex. So Alex, you told us about this amazing story from doing molecular research all the way to pharmacological and applications and cosmetic development. So tell us a little bit about the challenges along the way that you had, and maybe also how do you see your work progressing from here?
Yeah, so I mean, I think a lot of the challenges is quite a long project, right? It spanned 7, 8 years. So really, it's forming those relationships with industry. So there's a lot of brainstorming, a lot of conversation, a lot of proofs of principle that came before any of the work, and it kind of just built. You know, it started in silico, then we went on to cells, and so of course we had to build that work before we finally took it to patch testing. And you know, we had 2 different industrial partners here. We had Walgreens Boots Alliance that helped us in a few of the experiments, but also funded the research. And then we also had Sederma, who helped us when it came to synthesizing these peptides and looking at their stability and their penetration and whether they'll get through skin or not, and also sensitivity, whether they're safe to apply to people. So you know, the challenges really was trying to communicate between all of these different groups and collaborators, and to come together with a very clear, coherent hypothesis and project, and of course an output, which we're hoping to publish very soon.
Yes.
And next steps. Yeah, so I mean, I would really be interested in learning more about. So we know that these peptides have differential effects, but we don't really know yet how they're working. So what is the mechanism? Are they being metabolized, which is leading to a change? Are they binding to receptors? Is it a general damage signal, or is it a bit more nuanced? And that's something that I'm quite interested in, because if we can figure that out, then when it comes to predicting peptides that might lead to regeneration of tissue and skin, then that'll make things a lot easier. And of course, we're also looking at application of these peptides across multiple, you know, different scenarios as well. That would be interesting.
Yes, that was a little bit of my follow-up question. You know, are you thinking of some other applications, any particular connective tissue diseases where this may be of interest?
Yeah, I mean, we're in discussions with them at the moment. I think my primary focus now is just understanding it a bit more in skin before we take it anywhere else. But yeah, I think you make an important point, and it's something that we're thinking about.
Yes, thank you, Alex. I have one more question actually for Abhishek. So they're asking, when you mentioned fat burning in sarcopenia, is the primary target addressing sarcopenic obesity?
Sorry,
So when you mention about the idea of fat burning, Do you mean that the primary target may be also reducing sarcopenic obesity?
No. It's actually the opposite. It's the browning of the fat, and that's a bad fat in the body. So the interferon alpha and tumor necrosis alpha factor and interleukin 6, they actually act on the muscle, the muscles to cause myolysis, and also on the fat to cause lipolysis. But the fat they're acting on is the healthy fat, not the bad fat. They're increasing the brown fat of the body, which is not good for the body, and that leads to more visceral obesity. That means you have more visceral fat, and then you have more intramuscular fat along the muscle tissue, and that's not needed. So basically the muscle is shrinking, the brown fat around the muscle is increasing, and the lipolysis is happening into uh, the good fat, and that leads to sarcopenic obesity rather than reducing sarcopenic obesity.
Okay, so we've got a couple of more questions as well. Tamara, just a quick question for you. Have you covered any inflammatory markers in your model?
Inflammatory models, no. Unfortunately, not. I think we have done some inflammatory markers maybe, but we have focused mostly on metabolic and redox reprogramming, and we haven't examined the possible inflammation in obesity, since we do not believe that the obesity could be just having negative effects, and that the inflammation could be bad. So we didn't go into that.
Okay, Tamara. Question for Alex. So in your mass spec, Alex, did you have an ECM protein that was fragmented or whole length in the original sample? And if you see an increase in collagen 6 with age in skin from mass spec, would you know whether it was ROS fragmented?
We let the human durofibroblasts secrete and deposit their matrix, and we then collect it whole, we trypsinize it, and then we analyze it by mass spectrometry. So it's not pre-fragmented. It might be fragmented because of endogenous proteases or proteases that the cells release. But really, we're just looking at whether we see increases in or decreases in protein abundance and composition with response to these peptides. Collagen 6, I'd have to go back and check to see if collagen 6 as a specific marker was affected by these peptides. Yeah, I'm not sure.
Okay, and just a couple of last points for you also, Alex. Could you mention what type of AI you used in your research?
Yeah, sure. It was a bidirectional neural network machine learning algorithm that took into consideration both the secondary structure as well as the predicted cleavage sites based on so it's similar to the Prosper model, but it incorporates two-dimensional secondary structure as well as just the primary sequence. Yeah, so you can find this. So we published it back in 2021. The paper is predicting proteolysis in complex proteomes using deep learning. So you can find that, and the link to that web tool is there as well.
Okay, and the last question for you, Alex, is so do you know whether the effect of randomized peptide sequences on ECM production and how these sequences would function when part of a polypeptide chain?
Sorry, can you repeat the question? I couldn't really hear.
Okay, so the attendee is asking, do you have an idea of the effects of randomized peptide sequences on ECM production and how these sequences might function when part of a polypeptide chain?
No, so we haven't looked at randomized, and that's something that actually we would like to incorporate. And you know, that's part of the journey of understanding the mechanism. We know that we predicted these tetrapeptides that come from matrix, and they seem to work. But the idea is, if we were to change one of the amino acids to a different one along the sequence, does it have an effect? Is something that we would really be interested in finding out. Randomized might be a good way of figuring out what you know, what works and what doesn't. We have to be a little bit more targeted, I think, if we're looking at mechanism. Maybe change one amino acid at a time and see what the effect might be. Yeah.
Okay, thank you very much, Alex. Well, I think that concludes all our questions for today. So I'm going to thank everyone for attending this webinar and for speakers for their brilliant talks. And you can continue this conversation online by following BiochemSoc or PP Publishing on Twitter or X. So we just want to say that we welcome submissions for speakers to present their work in 2024 webinar series. If you've been inspired by our speakers today and would like the opportunity to present your research, then please submit an abstract for consideration by 22nd of February, and you can visit our website for more details. All of the upcoming webinars are listed on the website, and if you have missed any of our 80+ webinars we've run so far and you would like to watch them again, including the webinar today, please visit our website or the YouTube channel. The recording from today's webinar will be available to watch within the next couple of weeks. So we invite you to join on 6th of February at 2 p.m. for our next webinar entitled Process of a Product, Incorporating AI into Assessment, where our speakers will be exploring the capabilities of AI models and how to successfully adapt assessment strategies to enhance learning further. And finally, I would just like to highlight then, as it's more important than ever to stay connected and engaged with our fellow molecular bioscientists, we encourage you to join the Biochemical Society's community of researchers and specialists if you're not a member already, and take advantage of many benefits, including discounted registration fees for society courses and meetings, access to a wide range of grants and bursaries, summer studentships, personal online access to 2 journals, and much, much more. During my career, I've been a member of Biochemical Society for 14 years, and I've reaped benefits including support for attendances at different national and international conferences, stay connected bursaries for childcare support, and now taking an active role in the society as part of a committee membership. So please visit the website and find out more. So I want to thank everybody for a great webinar, and I say bye-bye for now.